So let's take a quick review of the overall structure of the muscle before we go into more details. So this is the muscle, that's the tendon, that's the bone, and the muscle consists of bundles of muscle fiber, and each muscle fiber bundle that you see is called a fascicle, okay, of muscle fascicle. And muscle fascicles are made up of muscle fiber, and muscle fiber in turn contains a lot of myofibrils. So this is where you have all the contractile filaments present. So a fetus contains approximately 50 myofibrils per muscle fiber. And in adults, we have approximately 2,000 myofibrils per muscle fiber, okay, in an untrained adult. Now, muscle fibers in turn contain sarcomeres. So these structures that you're seeing in here, these zigzag structures, right, from this blue line to this blue line, that is a sarcomere. A sarcomere is made up of myofilaments, and all the stuff that is required for contraction is present over here, mainly the actin and myosin. And these actin and myosins are myofilaments. So here you see a muscle fiber, and then you see the nuclei, that's the sarcolemma of the muscle. And inside the muscle fiber or a muscle cell, you see a whole bunch of myofibrils, which contain a whole bunch of different sarcomere units. So from here to here is a single sarcomere. So sarcomere is the functional unit of a muscle cell where contraction occurs. Okay, so compare the muscle fiber to pull and peel Twizzlers. So that if the Twizzlers, a pack of Twizzlers is a muscle fiber, each Twizzler is a muscle fibril. So if you peel a Twizzler, each string would be a myofilament containing a lot of sarcomeres. Okay, so sarcomeres contain neatly organized proteins, and the main ones are actin and myosin, which make up the myofilaments for contraction. Myofilament is basically all the proteins that make up the but the major ones include actin, myosin, and titin, which is an elastic filament. So the sarcomere is made up of a lot of proteins, but there are three major myofilaments. Okay, the thin filaments, thick filaments, and elastic filaments. The thin filaments are made up of actin. The thick filaments are made up of myosin, and the elastic filaments are titin that gives a slight elastic nature to allow proteins to pull during contraction. Okay, so the myosin looks like this. So there are a whole bunch of myosins in a myosin filament, and in the end it becomes a really thick filament. So it's a lot thicker than actin. So if you pick out one string of myosin, this is what it would look like. It looks like a golf club with a head over here. And the thin filament is known as actin. Actin additionally contains some regulatory proteins, troponin and tropomyosin. So in this picture, you see troponin over here. The string itself is tropomyosin. Okay, so if you remember, we talked about how calcium and ATP are really important in contraction, right? So troponin and tropomyosin basically act like a switch that determine when the fiber can and cannot contract. So this is where calcium comes in. When the troponin attaches to a calcium ion, it changes the conformation or the shape, and it pulls the tropomyosin filament away from these binding sites. Okay, so if you see in this picture, the binding sites are covered. So they're not available for attachment to myosin head. But in this picture, after calcium ions bind to troponin, the troponin pulls the tropomyosin away from the binding site, exposing the binding sites or the active sites or actin. Now the myosin will be able to attach to these active sites and pull on the actin filament to cause contraction. During any given contraction, a whole bunch of proteins, with a whole bunch of myosin heads attach to these binding sites to cause an attachment. So once the myosin head attaches to these binding sites, it's going to pull the actin filament to cause muscle contraction. Okay, so at any given time, you'll see a whole bunch of myosin heads attached to these binding sites like this. Okay, so for just a purpose of even understanding, I've shown just one head. From histology lecture, you should remember what a muscle looks like, a skeletal muscle fiber looks like. They have striation, you see peripheral nucleus, and then you see a dark and light striation. So the next picture, we'll actually talk about what these light and dark striation are and what makes up the striation. So just keep in mind, thin filaments are made up of actin and thick filaments are mainly made up of myosin. Okay, so this picture at the bottom of the page is an artist rendition of the actual micrographs that you see on top. So this is what you'd see if you magnify the striations a lot higher. And the I band, so we're just going to focus on the I band, A band, and the Z discs over here. Okay. So let's start with the I band. The I band is a part of the sarcomere that contains thin filaments. Okay, so this I band that you see over here is the lighter striation that you see under the microscope. So this is where the I band is. So this orange filament over here is the actin filament. This green filament over here is the elastic filament, which is titin. Okay, so the A band is this dark band over here. The A band is the area of overlap between the thin and the thick filaments. Okay, so in this picture over here, this area is the A band, which is a dark band under the microscope. Okay, the A band contains the overlap between the thick and thin filament. A single I band, you can see, spans two neighboring sarcomeres. Okay, and each sarcomere has a boundary known as the Z disc. Okay, Z lines or the Z disc attach neighboring sarcomeres together and it marks the boundary of a sarcomere. The thin filaments, which is right over here, so these thin filaments are attached to the Z lines on each end, but the thick filaments are held to the Z line with the help of elastic filaments, which are known as titin. Okay, titin. I think I should write that down over here. Just a sec. So let's quickly revise. Titin is the protein that makes up elastic filaments. I band is made up of mainly thin filaments. A band contains both thick and thin filaments because this is the region where both filaments overlap. The Z lines mark the boundary of a single sarcomere, and the H band, that's right over here, is known as the bare band. So H band is known as the bare zone because there's no overlap between the thick and filaments over here. And this line right in the middle of the sarcomere is called the M line, or the middle. You might be thinking, okay, how did they come up with this name? Basically, they just started pointing at structures and giving them random names. So there's no basis behind how they named these structures in a sarcomere. Okay. So hammer this in your head. Sarcomeres shorten to cause contraction, and muscles contract to pull. Muscles always pull, okay? And contraction literally means shortening. So during muscle contraction, notice this picture. During muscle contraction, the I bands shorten. So wait a minute. You'll see the I bands shortening, but the A bands remain the same. Okay, so I'm talking about the band, not the filaments. Okay, the length of the thick and thin filament do not change, but they rather just slide over each other to cause contraction. And when they slide over each other, the Z lines are brought closer to each other, right? So when you flex your biceps, these biceps basically contain at least 10,000 units of sarcomere, and when each of the sarcomere unit contract, it causes a muscle to pull or contract. And keep in mind, the length of the filaments do not change. The filaments merely just slide over each other to bring the Z disc closer together. And this is called the sliding filament model. Let's quickly review the different types of proteins in the muscle fiber. You have contractile proteins, actin and myosin. Myosins make up the thick filaments, actin make up the thin filaments. You have regulatory proteins on the actin, namely troponin and tropomyosin. Troponin and tropomyosin decide, well, they're like a switch, and decide when the muscle can contract. Okay, so troponin and tropomyosin are present on the actin, and the structural proteins are of various types: chitin, nebulin, alpha actin, myomesin. The one that I'm going to focus on is dystrophin. The reason I want to talk about dystrophin is because this is clinically important. So if you've heard of muscular dystrophy is a neuromuscular disorder, and the most common severely debilitating neuromuscular disorder is Duchenne muscular dystrophy. Now, Duchenne muscular dystrophy is also known as DMD, and it affects about 1 in 3,500 males. Why? Because the gene for this protein is located on the X chromosome. And males, remember, only have one X chromosome. So if they have a defective DMD gene on the X chromosome, they're basically the ones that will end up having a disorder of dystrophin, because this gene is located on the X chromosome. Okay. In the DMD gene, or the Duchenne muscular dystrophy gene, there's a change in the way dystrophin protein is made. And why is this protein important? It's because this protein is this red protein over here. It links the outermost myofilaments to the proteins and the layers of the endomysium. So it basically keeps the myofilaments held together with the sarcolemma. Okay, and any defect in dystrophin produces muscular dystrophy, and the most common type is Duchenne muscular dystrophy. Okay, and this is why, well, because it is present in the X chromosome, is why males are affected more than females. Without this gene, your body would make very little dystrophin, and without dystrophins, these muscles would become leaky, and it will cause the muscles to weaken. Okay. When the muscle cells weaken, they atrophy and eventually they die.